As Sarah said, my name's Ian Snapp. I'm from Ridgeway High School, and I did a neuroscience internship out of the Scripps DNC in San Diego, California. All right, I'll start with all the stuff that you're interested in, so lab maintenance. Gonna be honest, someone's got to do it, so you're gonna make the intern do it. Did a lot of the cleaning of the dishes, so using the dishwasher, and then the autoclave to sterilize everything. The whole lab uses it. The second half was a lot of the preparation of the common resources, so agar plates, LB broth, and TC water. Sterilize it in the autoclave, and then decanter it into any respecting containers afterwards. As for actual science, the part of the lab that I was in was mainly focused on metabolism within neurons. So it had been identified that PDH, an enzyme that manipulates metabolism, has a specific sequence of DNA that expresses it. We now know that sequence, we know the sequence of DNA, so we're able to manipulate it within cells. To do that, we use a PCR reaction, as previously mentioned, which essentially means taking double-stranded DNA, denaturing it, and ripping into single-stranded DNA, and then bonding what's called a primer, so a tiny piece of DNA on it, and extending it with heating, and then doing that over and over again to get a somewhat large sample of DNA with the manipulated sequence in it. That's the PCR machine, that's the general process, and then that's the pipettes and solutions used to actually prepare it. Once you have the sample DNA, you need to make a lot of it because you don't have a large enough quantity to actually do anything with to begin with. So we use bacteria, we transform it into bacteria, which essentially means you inject it inside a bacteria, and then as the bacteria is reproducing, it is also replicating a bunch of the DNA that you have. My bad. Once you have a bunch of the DNA, you pick colonies, so you just swab it from the agar it's growing on, put it into LB broth, a liquid form, and then isolate the DNA by dissolving away the bacteria and filtering out all of the other contaminants to leave behind DNA left. That's the, That's the mini prep. We use a centrifuge to actually filter everything out. That's LB broth. And once you have that, you have a small sample of isolated DNA that you can sequence to make sure you have the right sequence first off, so you actually manipulate the right thing during the PCR reaction. If you did, you move on to a larger quantity called a mini prep. So it's pretty much the exact same thing, but you don't use the centrifuge, and it's bigger. Once you finish isolating the DNA, you measure the concentration, and then you have a final concentration of isolated DNA with your manipulated sequence. You need to get that into some sort of mode of transportation to actually test it and analyze it. So we use a virus called adeno-associated virus, AAV. You put the sample DNA inside the virus, and then you can infect tissues with it, and it will express the sample DNA. First, we need to make it. So you put DNA of the virus and the sample DNA inside of human embryonic kidney cells. Long story short, they take up the surrounding DNA easily and can make a ton of viral capsids, so bodies, quickly. It's cheap and it's ethical enough to use in a lab at large scales. Once you have enough of that virus, you isolate it and in a similar process, digesting away the actual cell body and then purifying it so you have a constant concentration, using a centrifuge and known density solutions to settle it out into a single layer and filtering it off. Now you're at the final step. You have a virus with the DNA inside of it, which you can then infect neuron-like cancer cells. They're highly reproducible and mimic neurons enough to give some sort of pattern. So as these neuron-like cancer cells fire, they could be doing it at a different rate or taking up energy at a different rate from the manipulated DNA. If there's any positive correlation, we'll move on to primary cells, so real neurons that come from baby mice because they are similar enough to humans to test on. That's successful. You move on to living rats, so you inject the virus into their brain and do behavioral studies. Once those are completed, the rat is killed, and their brain is then suspended in a gel, frozen, and sliced into thin strips, which can be dyed so that you can highlight the areas that the virus was infected and expressed in. I mainly only worked on the first 3 steps because, I'm not a grad student. The last 2 steps involve a bit more brain power than I have at the moment. Outside of this, I lived alone in an Airbnb. Housing situation was a little complicated in San Diego, big city, not a lot of opportunities to find stuff. So rented out an Airbnb, lived alone in there for the 6 weeks, with the exception of my dad and my sister coming out on the way out to help sign me in, and my mom briefly visited just to see San Diego and what I was doing in the internship. A lot of traveling, visited the Grand Canyon and Sierra Nevadas, Vegas, a lot of things on the way out and back in particular. Bit more cuisine, or whatever, okay. Bit more food than like tacos and burgers. So, ate sushi, that's delicious. Don't get a lot of that in Colorado. Learned how to surf. And then, since the waves are a little less gnarly in Colorado, I learned how to skateboard. And a bunch of the city life kind of things like zoos and beaches and stuff. Reason to become a Pintern, figured enough people were giving advice. I would say that if you have a passion for learning, you're going to go into it on a lot more involved level than you would probably in your free time. It's going to be challenging to your comfort zones, whether it be socializing with people who speak Mandarin when you don't, or being several thousand miles away from home. You're going to expose yourself to new environments, and it's a test on a professional academic career, something that you're not going to get just being in high school, while you're going to get an education. And as a bonus, but not as primary reason, it is marketable to colleges and future jobs. I'll give a special thanks to Scripps, the staff of Ridgeway, and Pinhead.
Yay!